Retinal neurons derived from human stem cells are a promising source of replacement cells for regenerating damaged or diseased retinas. As scientists progress towards translation of cell therapies for restoring vision, they encounter challenges, including how to deliver the cells, ensure that they integrate appropriately with host tissue, and enable proper function after transplantation. Welcome to The Scientist Speaks, a podcast produced by the Scientist Creative Services team. Our podcast is by scientists and for scientists. Once a month, we bring you the stories behind newsworthy molecular biology research. This episode is brought to you by Bio-Rad. Bio-Rad is a global leader in developing, manufacturing, and marketing a broad range of innovative products for the life science research and clinical diagnostics markets. With a focus on quality and customer service for 70 years, their products advance the discovery process and improve healthcare. In this episode, Iris Kulbatski from The Scientist spoke with Deepak Lamba, a distinguished scientist in the Department of Immunology and Regenerative Medicine at Genentech and an associate adjunct professor at the University of California, San Francisco, to learn more about how advances in cell culture models and associated technologies help researchers progress towards retinal cell therapy.
Vision loss is a devastating condition. While many promising treatments exist for delaying degenerative eye disease processes, restoring lost vision by rescuing or regenerating damaged neurons is challenging. For Deepak Lamba, exploring novel cell therapies for restoring vision was personal. As a former emergency medicine doctor, he made the move to translational research after realizing that there were very few effective options for his father, who was diagnosed with glaucoma.
During my medical training, I got involved in ophthalmology. My father was diagnosed with glaucoma, which is this disease where the output neurons from the eye to the brain get affected. That was my first deep dive into really understanding. Unfortunately, over the years, none of the treatments ever worked on him. He went through multiple surgeries, multiple drugs, and nothing ever worked. He's now completely blind. That got me really interested in seeing what else is out there. And there wasn't that much. The current therapies just never worked on him. What else can I do as a doctor when I can't even help my own family? That's why I got into research.
As a graduate student, Lamba bioengineered retinal prosthetic devices before pursuing stem cell research. Lamba was fascinated by retinal developmental biology and was excited about the potential of using human stem cells to regenerate the vital neural connections that are required for vision. The concept that dysfunctional cells could be replaced to rescue lost vision was a grand idea at the time, because the human eye does not regenerate itself. Despite the robust progress of human stem cell therapy research for vision restoration, Lamba and his colleagues faced formidable translational challenges over the years. In addressing these challenges, the field experienced some foundational breakthroughs, which have brought scientists and patients closer to the reality of restoring lost vision.
My naive view when I first started this, I've made these cells, now we can fix it in patients. Then you realize all the challenges of going from an idea in an academic lab to something that actually will help patients in the long term. There are lots of challenges associated with that, figuring out how to make that into a process which is safe, how to make that into a scalable process to help multiple patients. Lots of these challenges that you don't really think about when you start coming up with an idea. How do you move from an idea to something that can help patients in the long run? The field has amazingly evolved. Two of the biggest advances, at least with relation to eyes, is first the discovery by Shinya Yamanaka of induced pluripotent stem cells. These cells that you can make from anybody, be it a normal individual or a patient, to have the ability to understand something in the eye. Most other tissues, liver, spleen, you could do a biopsy and understand what's going wrong. In the eye, there is no opportunity to do that. Animal models don't really represent the human disease case. This discovery completely transformed the field. The other work was by the late great Shiki Sasai, who moved the field from using two-dimensional tissues to these three-dimensional organoids and really getting to those intercellular, intertissue interactions, which are key to disease development, making better tissues for cell therapies. The next level is complex in vitro systems where you can have multiple tissues interacting and getting you a better insight on how you can try to fix things.
Lamba's academic research has focused on two broad areas. The first has been on stem cell therapies, how to grow the right types of cells, how to scale the process, and how to optimize the ability of the cells to integrate into host tissue and support better neural rescue. Lamba likens the importance of optimizing the transplantation environment to choosing the best soil for a garden. While the seeds may be high quality, providing subpar soil conditions will negatively affect plant growth. Similarly, cultured stem cells may be highly viable in vitro, but their ability to rescue vision after transplantation may be undermined by inadequate characteristics of the local tissue environment after transplantation, such as an inhospitable immune environment.
We worked a lot on showing proof of principle that we can rescue using these cells that we grow in the lab. We have a couple of papers on taking these cells, transplanting in mice which are born completely blind from birth. We could rescue vision. And then we looked at why this rescue is somewhat limited, nowhere close to what a normal vision would be. One of the biggest challenges in the eye is immune rejection. These cells, you can transplant them, but they aren't the patient's cells. So your body recognizes this as foreign. We went in looking at affecting the interleukin 2 pathway, which is critically involved in these natural killer responses to get rid of foreign bodies. And there we could improve rescue of close to tenfold. We've continued to work along those ideas to see if rescue can be further enhanced by creating a more receptive immune environment. In conjugation with our cell therapies, we could show a massive improvement in integration and functional improvement in these mouse models. Slowly building up to get the best cells and the best environment, then test that in humans.
The second focus of Lamba's research has been on how to use complex human stem cell cultures as model systems to understand disease processes and inform the development of better therapeutics. For example, Lamba's team used induced pluripotent stem cells from patients with congenital retinal degeneration and vision loss to grow retinal organoids, examine the cellular and molecular basis of disease processes, and pursue genome editing strategies to eliminate mutated proteins that contribute to disease. This work provided an important proof of principle for future patient trials.
One of the big papers that we published 3 years ago was on modeling patients with mutations in a gene called CRX. These patients have very early onset vision loss. Stem cells from patients with different mutations in different parts of the same protein had different phenotypes. Those phenotypes matched the patient's phenotypes. For example, one set of mutations affected certain color vision, and that matched very closely with the type of cells that were born in these mini organoids. Even though the patient mutation was because of just one of the two alleles of the DNA having the mutation, the dominant mutant protein was overtaking the function of the recessive or the normal protein. We could get rid of the dominant negative protein and rescue in an organoid setting all of the phenotypes. Gene replacement is a lot more challenging, but knocking things down is much easier.
Improving cell delivery strategies is also on Lamba's radar. The anatomy of the human eye makes transplanting cells into ocular spaces a formidable challenge. The proximity of different cell layers that are responsible for different aspects of vision makes the risk of collateral damage high. This is particularly evident in disorders like age-related macular degeneration, in which two types of visual cells are lost from the subretinal space in the back of the eye: light-sensing photoreceptor cells and supportive pigment epithelial cells. Safely delivering cells to this space is difficult and risky because of the potential damage such a surgical procedure can cause to the rest of the eye. Moreover, supporting the cells in their respective jobs once they are delivered is necessary for proper functional integration.
Recently, here at Genentech, we've been looking into devices where we don't have to go through the eye to deliver this. We go from behind the eye, which is the approach that we took in a partnership with Lineage Therapeutics. If you go from behind the eye and deliver it to that space, the corresponding damage is much less. Getting the cells to connect or support the cell types that you need to support is a bigger challenge when it comes to light sensing cells. The pigment epithelium is a support layer, so as long as you have the right cells in the right space, they should be able to support the light sensing cells. But when it comes to the photoreceptor side of things, you don't want them to assemble amongst themselves. You want them to connect with the host eye make the right connections, make the right synapses. And those are significant challenges on moving from an idea in a lab to what would happen in a patient. That said, our brain's actually amazing. We have lots of really good examples, including patients who've never seen anything, and even much later in life, when you rescue vision in those patients, the brain starts making sense of these new inputs. The brain is very plastic. Even if you can rescue some amount of vision, you will be able to give some useful vision in these patients who are at the end stage of disease.
The question of what proportion of cellular rescue needs to occur in order to restore reasonable vision is a difficult one to answer. Lamba noted that patients can lose nearly 80% of their vision before realizing the extent of the loss because of the brain's inherent plasticity. According to Lamba, a common approach is to aim at rescuing predominantly the cone cells responsible for central vision, which is most often used for day-to-day activities like navigating around obstacles or crossing the threshold of a doorway safely. Lamba estimates that approximately a few million photoreceptors would be required to rescue enough central vision to make a difference to patients, which is quite feasible in terms of scalability.
The idea is, how do we get better at cell therapies? Genentech is involved with this effort to get stem cell therapy with relation to the pigment epithelium, the support layer, to the clinic, and figuring out challenges, better cells and better processes to get those cells made so we could treat thousands of patients who need these therapies. We're working very hard on that. On the flip side, now that we can build these complex systems and have the depth of insight using single cell technologies, we're trying to put that together, mostly focusing around diseases affecting large populations, age-related macular degeneration, glaucoma. Both of these are huge challenges. There are millions of patients who have these disorders around the world. We have an amazing team up here where we can lean on people who can think through how to make the right drugs to match your hypothesis of a pathway that's dysfunctional to how do you think about getting that to the patients. The dry form of macular degeneration and late stages of glaucoma, there isn't that much we can do.
Lamba is hopeful about the research progress in the field and the incremental discoveries and cutting-edge techniques that pave the way for tangible translational progress. Lamba notes that age-related diseases like macular degeneration are difficult to model in vitro using stem cells because of their inherent immaturity. Moreover, there is no single gene or molecular pathway that has been deemed causative. The complexity of the system is likely related to age-related abnormalities in cells and their microenvironment. Lamba is currently investigating human disease post-mortem tissue to gain deeper insight at a single-cell level into such aspects in a way that is contextualized to patients and future targeted therapies.
Single cell analysis, you can go cell by cell, understand not only the transcriptome, what genes are changing, but also the epigenetic regulations happening within these cells. There's a technique called ataxia. You can do multiple omics analysis where you can look at both how the gene expression is changing, what part of the gene or our DNA is open or closed, and start making predictions based on that. We've now gotten a much better insight on why a certain mutation did that and a certain other mutation did something completely different. Combining all of these and human cell from a patient, their own mini retina in a dish, and now ability to look at things at a single cell level just has really revolutionized how we can get into the depths of a lot of these diseases and make better predictive hypotheses.
How far are we then from a tangible, clinically relevant retinal cell therapy? Multiple clinical trials for rescuing various types of cells in the eye are currently underway, including Lamba's own work at Genentech. Lamba believes that the future of these efforts is bright, despite the inherent challenges.
We've moved the needle a lot over the last decade or so, from knowing how to make these cells to figuring out the staging of these cells and the microenvironment they would go into and what needs to be done there. Something like this requires a big team approach.
Thank you for listening to The Scientist Speaks. This episode was produced by the Creative Services team for The Scientist and narrated by Iris Kulbatski. Thanks again to Bio-Rad for sponsoring this episode. Please join us again in December as we learn how researchers seek solutions to tumor treatment resistance. To keep up to date with this podcast, follow The Scientist on social media and subscribe wherever you get your podcasts.